Well, did Pfizer and BioNTech falsify key data in their COVID tests for their vaccines, essentially trying to lie to all of us about what was actually happening with these results? That's the finding of investigative journalist Sonia Elijah, who's written extensively on Pfizer and BioNTech and their cover-up in all of this. Journalist Sonia Elijah joins me now. Nice to see you. Welcome to the show.
Thank you so much, Clayton, for having me on.
Well, it's my pleasure. So thank you for reporting on this and bringing this to light. What did you find when you were going through these new documents and results and tests that were starting to come to light? What did you find?
Yes, so I actually wanted just to put this into context. Now, I wrote a two-part investigative report for Trial Site News last year. This was on the leaked European Medicine Agency emails and Pfizer-related documents. And it related to the time period of mid-November 2020, running up to the time when emergency use authorization was granted to the Pfizer and BioNTech vaccine. What was really alarming with these emails and the documents is that the regulators were highly concerned about the quality of the vaccine. And as particularly what they had noticed was there was a significant drop in the RNA integrity level of the commercial batches of the vaccine. These were the batches done sort of large scale, large volume. It dropped down to 55%, the RNA integrity level, compared to 78% in the clinical batches, which were the batches used in the clinical trials. So what the RNA level integrity, sort of what it points to, it's how intact the mRNA molecule is in the vaccine. And we know, looking at the documents, a relatively intact molecule was needed for the efficacy, for safety. So what they found were these truncated, fragmented mRNA species in the vaccine. And what was shocking is that they literally dropped, the way they sort of solved the problem was to drop the criteria, the standard, down to 50%. To allow at least half the mRNA molecules to be truncated. And they had no idea, this is the regulators and the vaccine manufacturers, how the safety and efficacy implications of the fragmented RNA.
So not to interrupt you, but they, being the regulators and Pfizer working in tandem, lowered the standard.
Down to 50%, so allowing half to be truncated. Now, where my recent report comes in, now, conditional marketing authorization was granted by the European Medicine Agency. They granted Pfizer and BioNTech this, the CMA was given on the basis that BioNTech, which is the marketing authorization holder, meet specific obligations because there was a concern with the quality of the vaccine. And one of those specific obligations, they sort of categorized it as SO1, and I'll just read it. There's a lack of experimental data on the truncated RNA and expressed proteins does not permit a definite conclusion and needs to be further characterized. Therefore, additional characterization data remain to be provided as a specific obligation. Now, following that, BioNTech had, in order to meet that obligation, they did a test called a Western blot. Now, a Western blot is a technique used to identify and separate specific proteins from a protein mixture extracted from cells, and it's based on their molecular weights.
And we have here from your piece, I want to put this on the screen that we'll be able to show our audience, a normal blot test. So can you describe, this is a normal blot test. What are we seeing in this normal blot test slide here?
So a normal blot test, you'll have the bands, you'll see these sort of black bands, and they sort of have an arc to it, almost like a slight smile. And there's smears you can see. They're done by hand, they're done manually. Now, what the Western blot that was provided by BioNTech to the regulator were completely regular, perfectly regular bands, all sort of uniform. And when you zoomed in on one of these bands, you could see perfect row of pixels. Now, a scientific expert provided me with further evidence. So what they did, they used a image analysis software to quantify the bands. And when they did that, they did a plot profile of the bands. And this is what is so alarming and so concerning, is you can actually see certain bands copied and pasted. So they've been literally duplicated in groups of 2 or 3. And that's what my report shows. And this is the new evidence. This is, I believe, the smoking gun, because it's a copy and pasted data. And what is just astounding at Clayton is that the regulators accepted these as authentic Western blots. Now, my report goes into what a conventional one looks like. Now, the counter argument is that all these are maybe automated. These are done by computer. Now, if you take that argument and run with it, it fails to explain the duplication of the results, because an automated Western blot will you'll still see variations in the positioning of the bands and their intensity. And it also fails to explain the terminology used by BioNTech and Pfizer. They use the same terminology that you would describe a conventional Western blot, one done by hand. Wow. So that argument just fails in my eyes.
Other pieces of your report where you talk about the major objections and the comparability, and you talk about the in this piece, and part of this, a major objection is the truncated and modified RNA species should be regarded as product-related impurities. What does that mean? That means there's a problem with these and these should not be allowed to go to market?
It means that they don't know what they are. There's no characterization data. It means that they don't know what the proteins are expressed by these truncated mRNA. So mRNA carries the code for the protein expression. So they have no idea what these proteins are, the expressed proteins are, right? And we don't know. Yeah, exactly. That's completely foreign. The BioNTech and Pfizer have failed to meet, even though it's astounding that the regulator says this specific obligation has now been fulfilled, but it really hasn't. They have done fraudulent tests in many scientific experts have looked at these, and it doesn't explain what these truncated mRNA, what the protein's been expressed by it, and we have no idea how the safety implications of those truncated mRNA species are, these product-related impurities.
How common is it for regulators and pharmaceutical industry reps, coming from Pfizer and BioNTech, to collaborate and to lower the standards by which we would get access to certain vaccines or medications? Does this happen all the time?
I would say this is unprecedented. I would say in this the large scale, the impact of this vaccinating. Right. We think about the billions of doses that have been administered, the boosters, and all of that. This is unprecedented. And we know that these regulators have been captured, right? We know that the bulk of their funding comes from the application fees that pharmaceutical companies pay to the regulator. And these regulators are supposed to be the watchdog. But in a sense, the watchdog's teeth have been pulled out.
So we know that this is England's. We're not England. We're specifically talking about the European Union on this. England just left the European Union.
Also the FDA. So the BioNTech provided the same Western blots to the FDA because the FDA had queries, and my report does comment on the.
So that's I know. Yeah, that's what I was going to get to on the FDA side. So now there's a history here, and then that's cataloged in Beth Macy's book, Dope Sick. And I want to just pull up a quote out of Dope Sick, if you'll allow me, where she writes, "It would not come to light for 9 more years that FDA regulators and big pharma executives had been quietly holding private meetings at expensive hotels at least annually since 2002 through a drug industry funded nonprofit, an ethical quandary, a Milwaukee Journal Sentinel reporter shone the light on in 2013. The meetings led to the development of enriched enrollment, an aptly named practice that allowed drug companies to weed out people from their studies who didn't respond well to their drugs, therefore tipping the balance towards FDA approval of new drugs and away from science. So in this situation, she's talking about how they were able to take people out of the studies that didn't respond well to the drugs and therefore tip the balance. In this case, it's different, right? They're using these tests and they're manipulating the test away because they're really not looking at actually human trials in this, are they? They're using fake data.
It's fake data, but also we know in the clinical trials, they did remove people from the trials. I've also looked at the Pfizer clinical trials, and I've written extensively on this for Trial Site News. I found anomalies, errors, just really shocking what I found. I've also interviewed Brooke Jackson, the whistleblower, and I found a third death in the clinical trial that was part of the pivotal, it was before the cutoff date, that the New England Journal of Medicine, it was supposed to go in that pivotal trial, but it was missing. They said there were only 2 deaths. I found a third. That's an increase of 50%. And I have written to, again, I've written to, as a writer, response to, the regulators and of BioNTech, Pfizer, sort of no response there. But we know that there's a lot of fraudulent activity has gone on because we're seeing the evidence.
Have any lawmakers come forward to say, thank you for this, we're going to look more deeply into this, or has there been radio silence?
Just now some people are reaching out to me, but yeah, it's been pretty much radio silence. It's obviously alternative media like yourself who are wanting to interview me on this, but mainstream media, as we know, will not report.
So they continue. Now, where are we sitting with these booster shots? These are in this lineup. We heard Australia. We reported yesterday on the show is pushing for a 5th dose now in Australia. So are we led to believe then that the same batch of booster shots that exist, where they're continuing to push people to go and get these booster shots and updates, and even trying to push now for an annualized version of this shot, we can expect the sort of same results here? Are we talking about the same batch of data, the same falsified documents that led to these now annualized shots and extended boosters program?
Yes, I believe that the regulators have no idea about the quality of these vaccines. They are going on what Pfizer and BioNTech are saying on their data. But we know the data has been compromised. The data has been, obviously my report shows has been copied and pasted, duplicated, and look highly unusual. Nothing like a conventional Western blot. What those results should look like. And it's very alarming because what are the safety implications of this? We know there's, we know there's a huge underreporting going on. Obviously, you look at VAERS, and that still is, we're seeing unprecedented levels of adverse events occurring. But you have to take into account this is severely underreported, right. Doesn't accurately reflect the actual harms that are being done.
Unbelievable. And finally, now we're starting to hear, and we're getting reports, and people are able to go to social media and share these stories. And we're opening the floodgates here on this. And I hope that there's an investigation. This is criminal. I hope there's a large scale investigation. I hope there's a massive trial of some kind. People saying, is it Nuremberg 2.0? Where we're actually going to get to the heart of this. I hope that there are brave enough lawmakers out there to hold these people to account. Sonia, thank you so much for joining us, bringing this information to light, and we'd love to have you back as you uncover more of this story.
Thank you so much. Thanks, Clayton.
Thank you. And we'll have links to Sonia's piece, her article in the description. So I encourage all of you guys to go and check and read that and her great reporting on this. And we'll have her back when we have more reporting as this unfolds. Unbelievable story.
Yeah, her demeanor was like, of course, the mainstream media is not interested in this.
Right.
It's become just the way that we greet each other, people who are willing to read now.
Well, and I love that she's just based on the facts. She's like, I don't need to be dramatic about it. I don't need to add any drama into this. I'm just presenting you with the facts. Like, they're literally fudging the data, and then we're getting stuck with these jabs. Like, this is unbelievable. Thank you so much for watching this segment here at Redacted. We are live every day at 4 p.m. Eastern time, trying to share the stories that the mainstream media will not cover. You should also come over and join our community of Redacted Rebels over at redacted.inc. That's our private locals community where we can share exclusive content that we simply cannot share here on YouTube. Come over and join the rebellion together right now by going to redacted.inc. We'll see you next time.